Okay, we're going to talk about chapter 5, which is on molecular genetics and tissue differentiation. Now, it turns out that the entire genome is made up of about 25,000 genes. And genes are basically these invisible information coding elements. Now, they're invisible to the unaided eye, but with certain microscopes and things like that, you can see the DNA which forms genes. Now, these genes exist within DNA in cells, and most of our genes are located within the nucleus of our cells. However, there are some genes within mitochondria. Now, what happens is that these genes in DNA are passed on to daughter cells when the cell divides, because what can happen is we can copy our DNA genomes and then pass that copy of the genes or DNA into the next cell when it divides. Now, DNA, or deoxyribonucleic acid, are these long chains of stable molecules that store our genetic information. Now, it turns out that the 25,000 genes or so that's contained within our genome are all encoded by a series of 4 different molecules, which are nucleotides. We have adenine, cytosine, guanine, and thymine. And these nucleotides, in a certain sequence or pattern, is what encodes our genes, or for our DNA. And it only takes about 25,000 genes to make a human. And each of these genes, each 25,000 of them, encodes for a specific protein. Now, in terms of our entire genome, we have about 46 long double-stranded chains of nucleotides, which we call chromosomes. So humans have 46 chromosomes. Now, the thing about this, though, is that they're not 46 unique chromosomes. It's 46 chromosomes total, but humans only have 23 different or unique chromosomes. The thing is that we have 2 copies of each chromosome. Because we have 2 copies of our 23 chromosomes, that gives us 46 total in a typical human cell. Now, it turns out that our DNA is made up of our 5-carbon sugar, which is our deoxyribose. It's linked up by a phosphate group. We call this the phosphate backbone. And then we have 1 to 4 nucleotide bases that are attached to our 5-carbon sugar, which makes our nucleotide. So if we look at this, at the molecular level, we have our phosphate backbone here. We have our 5-carbon chain ribose sugar. And then that's hooked up with all of our nucleotides, which we have adenine, cytosine, guanine, and thymine, or A, C, G, and T. Now, it's the sequence of these nucleotides that really contains the information within our DNA. And there's these millions of links or chains of these nucleotides that makes up a particular chromosome. Now, what happens is that we talk about DNA as being a polymer, and polymers are basically just long chains of some sort of molecule. Now, because our DNA is made of nucleotides, we call DNA basically a nucleotide polymer. And there's 2 types of nucleotides. We have the pyrimidines and the purines. The pyrimidines are cytosine, thymine. The purines are guanine and adenine. And it turns out that based on their sizes, the pyrimidines can only hook up with other purines, and purines can only hook up with other pyrimidines, because purines are basically going to be not the right size to hook up with other purines. So what that means then is that cytosine always links up with guanine, and thymine always links up with adenine. I'm talking about in the double helix structure of DNA. Now, our phosphate backbone is actually hooked up with these phosphodiester bonds, which are pretty strong, and so they're more difficult to break. And so our nucleotides here that occur like a repeating sequence, those are attached to that phosphate chain, which we saw in the previous slide. But these nucleotides are linked up together across the DNA double helix through what are called hydrogen bonds, which are a little bit more weak. So if we look at the DNA double helix here, here we have the phosphate backbone on one side, phosphate backbone on another side, and then here's our nucleotides. So we have adenine, thymine, guanine, and cytosine. And if you look at these little red dots here, what these red dots indicate is the type of bond that holds these nucleotides together, which is basically a hydrogen bond. So we have 1, 2 hydrogen bonds between adenine and thymine, and then 3 hydrogen bonds between guanine and cytosine. Now, it's the hydrogen bonds here that basically hold the DNA strands together. However, these hydrogen bonds are weaker than other types of bonds like covalent bonds. So these other solid lines here indicate covalent bonds, which are stronger. So because these hydrogen bonds are weaker, it allows for the strands of DNA to separate, and these strands of DNA need to be able to separate during DNA replication. Now, these hydrogen bonds are weaker, but they hold the strands of DNA together fairly tightly. However, these bonds can be broken with heat and enzymes. Now, remember, guanine always binds to cytosine, adenine always binds to thymine. So we talk about these nucleotides as having complementarity, or basically they complement each other. So guanine always complements cytosine, adenine always complements thymine. Now, if we look at the DNA double helix here, it kind of winds around this helical structure, and we can see that the nucleotides here, cytosine and guanine, hook up, thymine and adenine also link up together. And it's the structure here that basically is what forms our DNA. And if you look at this, it kind of looks like a zipper. And so we talk about the strands of DNA being able to unzip, and that way you can copy each strand of DNA before the cell divides. We talk about the DNA as having a double helix structure, which kind of looks like a ladder. And so the base pairs are basically nucleotides of the rungs, and it makes a turn about every 10 pairs or so, which is more at the microscopic level. Now, DNA is actually packaged within the nucleus, so it's not just sort of loosely floating around inside the cell. Instead, what we have is the DNA double helix, and it coils around itself, as well as coils around these little proteins called histones. Now, these histone proteins are basically DNA packaging proteins, and what they serve as is basically a scaffolding for DNA to wrap around. And when we have lots of histones together, this forms what's called a nucleosome. And what happens is that once these histones start to package DNA tightly, it actually starts to coil in around itself, which forms a supercoiled structure, which then eventually can be packaged into something that resembles an entire chromosome. Now, this condensed chromosome really you only see during mitosis when the cell's dividing. So especially during metaphase when the chromosomes start to line up at the metaphase plate. Otherwise, what happens is that these chromatin, which is basically DNA and all of its histones, is actually more of its uncoiled state. It's kind of more just sort of spread throughout the nucleus, not really condensed. However, before the cell divides, these chromosomes can condense into something that looks like an X. Now, we talk about DNA as having a sort of central dogma. And the central dogma of DNA replication is that DNA can be copied into more DNA. We call this DNA replication. However, DNA can be also copied into a more transportable form called RNA. And the process of converting DNA into RNA is called transcription, because you're changing the script from DNA to RNA. And we can also we can also copy RNA into protein. We call this translation, because you're actually changing the type of molecule here from RNA into a different type called protein. So basically what we see here is that from DNA, we can copy that into a more transportable form via transcription into RNA. RNA can be copied into protein via translation. This occurs at the ribosome until we get our end protein. And that protein could be like for cell structure, support, defense. It could even be an enzyme that's involved in metabolism in the cell. Now, one important note too is that there's another process called reverse transcription. Now, reverse transcription is basically where you can take RNA and turn that into DNA. Now, this typically doesn't happen in a normal human cell. Reverse transcription is a process that some viruses use to insert their genomes into the host cell genome, to kind of hide from the immune system or go dormant. But reverse transcription is converting RNA into DNA. So DNA replication begins when the cell starts to prepare for division. And what happens is the DNA double helix will separate by breaking those hydrogen bonds that hold each of the strands together. So you're basically separating the nucleotides, like separating adenine from thymine and guanine from cytosine. What happens is that replication enzymes basically help direct the replication of those strands. So that what happens is that once the DNA unwinds or unzips, we have replication enzymes like DNA polymerase, which comes in and copies each strand. So what you end up with then are two identical copies of the genome now. So basically a copy of the chromosome. Now, what this looks like then is we have an unzipping of the original DNA strand here, and we can see that this enzyme, DNA polymerase, is coming through and making a copy of each of these strands, so that from one chromosome with two strands, you can get two chromosomes with four strands. Now, what this is showing then is basically this process of replication. And so what this is also kind of trying to tell us is that there's complementarity in every time you replicate it. And so what we can really determine then is that as long as the strands are copied with high fidelity, or sort of, copied precisely, this original parent double helix should be identical to the daughter double helixes coming down the line after 2 rounds of cell division. That's just assuming that there's no mutations that occurred during replication. Now, what happens, you guys, is that our genes all direct the synthesis of some sort of protein. So we talked about how there's 25,000 genes in our genome, and every single gene directs the synthesis of some type of protein that's necessary for our cells to survive. What happens is that these proteins are comprised of basically just different chains of amino acids. And what's important to note is that once these chains of amino acids are formed after translation, they can fold into a complex 3-dimensional structure, and the structure of that protein really tells us about the function of that protein. Now, some proteins have a structure that's important for basically support for the cell. Other proteins have structures that help provide an enzymatic function for that protein. It can basically help chemical reactions speed up. Now, there's about 20 different types of amino acids, and these amino acids basically can join in specific sequences to form a specific protein. So when we talk about the shape of a protein, it's really determined by the sequence of amino acids, or sequences of these biomolecules that can really come together to form that 3-dimensional shape. Now, information is contained in 4 kinds of DNA nucleotide bases, and those actually code for 20 different amino acids. What happens is that the sequence of nucleotides in DNA come in sequences of 3, and those sequences of 3 nucleotides we call triplets form what's called a codon, and each of those code for the 20 different amino acids. So a certain sequence of nucleotides, whether it's like ACA or AGT, all of those sequences encode for a specific amino acid. Now what that means then is if you look at the entire genome, or let's just say an entire gene, the sequences of nucleotides going across that gene in sequences of 3 going across the entire length, each of those 3 nucleotides encodes for a specific amino acid. So what that means then is that we'll have amino acids in a specific sequence because that relates to the sequence of nucleotides on our DNA. What happens then, you guys, is that DNA gets copied into RNA, which is more of a transportable form of the gene. That RNA can travel on over to a ribosome, where the RNA is then converted to protein sequence, basically amino acids coming together to make protein. RNA is very similar to DNA, except for some key differences. For one, RNA is typically single-stranded molecule, whereas DNA is usually a double-stranded molecule. Also in RNA, we don't find any thymine. Instead, we have something called uracil, which is another type of nucleotide that's unique to RNA. What's also important to note, too, is that the sugar molecule backbone is a different type of sugar than DNA. It's ribose, not deoxyribose. Now, there's different types of RNA. We have ribosomal, messenger, and transfer. Ribosomal RNA is made by the nucleolus, and this is a type of RNA that basically works in conjunction with the ribosome to help make protein. We have something called transfer RNA that's also primed in the nucleolus, and this is what helps carry amino acids to the ribosome. And messenger RNA is actually what's being copied from DNA. So when we talk about copying DNA into RNA, that's the transportable form of the gene, which we call messenger RNA. And really what it does is it brings the message to the ribosome to direct the synthesis of that protein from the gene. Now, transcription is the process of converting mRNA into protein, but really this mRNA initially comes from the DNA template. So we have our DNA gene, which is copied into mRNA. That mRNA can leave the nucleus, head on over to a ribosome, where then that can be basically converted to protein. Now, transcription is just the process of converting DNA into mRNA. And this gene is transcribed when something called RNA polymerase copies DNA into messenger RNA. Now, what happens then is this RNA polymerase basically can separate your DNA double helix, and it can convert DNA into messenger RNA. And messenger RNA is complementary to that DNA genome in the sense that it has sort of the same genetic sequence. It's going to encode for the same series of amino acids. So what this looks like then is that our DNA double helix can unzip, our RNA polymerase can come in here and start to copy our DNA sequence into messenger RNA, which is a more transportable and smaller form of the gene. Now, translation is the process of converting messenger RNA into protein, and this occurs at the ribosome. Now, messenger RNA is read linearly from one end to the other, and it's read in a sequence of 3 nucleotides. Remember, each 3 nucleotides along the length of that messenger RNA encodes a specific codon, which encodes for a specific amino acid. So what that means is that since we have long sequences of 3 nucleotides on an entire length of messenger RNA, each of those 3 nucleotides is a codon, which directs the specific amino acid on transfer RNA to bind and form a protein. So what this looks like then is that our transfer RNA brings in a specific amino acid that has a specific anticodon which binds to the codon sequence of messenger RNA. So really the ribosome is just the assembly site for protein because messenger RNA brings in the recipe or the sequence. Transfer RNA brings in the basically like the components of the recipe, and the ribosome is like the kitchen which kind of brings it all together to make your protein. So what happens is that messenger RNA will start to line up with your tRNA molecules, and the ribosome will help catalyze the peptide bonds between all the amino acids to start forming your protein chain. Now, what happens, you guys, is that basically our messenger RNA serves as the template for directing the sequence of protein. tRNA will move along the mRNA sequence and start to bind, and then the ribosome will basically help stick those amino acids together in a specific sequence that correlates with the sequence of messenger RNA. So what that means then is this newly synthesized protein is similar to the sequence of messenger RNA from which it was directed by. Just kind of tying this whole process together, basically what we see here is this messenger RNA. Remember, a sequence of 3 nucleotides of messenger RNA forms a codon, and then tRNA has an anticodon, which can bind to the specific codon. Remember, tRNA brings in an amino acid, so when tRNA binds to messenger RNA, it can actually link up with other tRNA molecules nearby to form a growing polypeptide chain. What we see is all these little spheres here is a specific amino acid, and this comes in a specific sequence, which over time will fold into a specific protein, which could be anything, like it could be a microtubule, it could be a specific enzyme that can be involved in cellular metabolism. Now, what's important to note is that not all of the genes in your cells are expressed simultaneously. Instead, what happens is that only specific genes are expressed at any moment in time. So not all 25,000 genes are expressed in a given cell. And this is really what makes cells different from each other. You know, although a brain cell will have the same genes as a liver cell, their functions are very different because brain cells express different genes than other cells. So what that means is that some genes are actively transcribed, other genes remain sort of quiet or quiescent, in that cells can actually change their expression based on the needs placed on that cell.